So thank you very much for this very nice introduction. And thank you all for coming to this plenary talk, and also the organizers for inviting me to this wonderful location, this wonderful venue, and hoping to interact with you also during the talk or after the talk or during tomorrow's workshop. I would also like to invite you, if you would have already questions during the talk, please go ahead and raise your hand. We can discuss them, I think. And you can also reserve them or take them for the last bit of the 1.5 hours that is planned for this session. So indeed, I'm Bart Rutten. I'm a professor in neuroscience of mental illness at Maastricht University in the Netherlands. And the title of my talk is The Inside Out of the Outside Within, reflecting on how the environment may be imprinted at the molecular level within the cells, and how we are trying to investigate these processes and bring these things inside out. So I'll discuss these few things during the next hour or so. And I also want to give a disclaimer, since I do have some unpublished findings in this, and you will learn about these. They won't be in the PDFs that will be circulated around, I think. I also want to invite you to think about, could we join forces, and somehow that you as a clinician or scientist here in Turkey or the different countries in the world would like to team up with our group in Maastricht or with the international networks that we are collaborating with. Please contact us. I think that's very important in this area of research. And I also want to make the disclaimer that the topics of my talk don't follow a very logical sequence, in the sense that there's one topic being discussed. It follows a personal trajectory, in the sense that I've started on this type of work for, say, 10, 12 years ago. And along this way, I envisioned that I wanted to do more and more research on the molecular biology of gene-environment interactions. And I started doing my PhD on the field of dementia. So therefore, my first work that we also discuss will be on dementia, and thereafter I will also touch upon other areas in psychiatry, being post-traumatic stress disorder and also on depression. Very briefly, but I won't go into detail, we may also discuss psychosis, but it's something that my colleague Dr. Sinan Guluxus, also here in the audience, will discuss tomorrow during a talk. So just about our center, let me see where the laser is. Probably you don't know where the city of Maastricht is located, so I just wanted to give you some background information. It's in the southern tip of the Netherlands, over here. It has a very nice university medical center, but the university is very young. It's only initiated in 1975, and it's one of the best young universities worldwide. So it has a very nice, innovative atmosphere, and it's located in a very nice, tiny village with only about 200,000 inhabitants, of which 30% is a student. So my own trajectory is that I did my PhD project in the years 2000 to 2004 on aging and Alzheimer's disease. And I was a medical doctorate, and then I embarked on my PhD project. And I thought this would be a very nice, purely biological-driven research project, which I investigated the processes behind neuronal loss in dementia, and investigating things like amyloid-containing plaques, the inflammatory response to this, sorry for this, and also how synaptic contacts would be disturbed and also be lost in the process of dementia. I also thought I was a very young scientist, and I was very keen to work on this area, but I found out that I think you need more and more people to collaborate with. You really need a group of lions that is working together with you to really get the work done. And along the way, I passed my research themes to more impact on, or more emphasis on gene-environment interactions, and thereafter epigenetics in the fields of dementia, psychosis, and PTSD. And the general framework is that what we're trying to do, and what we learn also in the textbooks, is that certain genetic factors or certain environmental factors, they increase risk for a certain phenotype. But I think the most challenging aspect is that if you would find in epidemiology a statistical interaction, the next step would be to go into more biology and biological research and try to find out at which level can we find this interaction, and how does it work? And I think only a handful of studies are really truly working not only on one specific layer of investigations, but are trying to combine this. I think Professor Carol Tominga's talk just an hour ago is a very nice example on how to try and to integrate this from the more symptom dimensions, transdiagnostic work, to intermediate phenotypes that then can be traced at the biological roots. So we're working in Maastricht with a very nice set of people, and I want to particularly also emphasize two people that we collaborate with. This is a psychiatric epidemiologist, Professor Jim van Os, who is working with us and now moved to Utrecht, but still connected to our research team, and Dr. Sinan Guluxus, who is really driving the field of gene-environment interaction research in our European efforts. This is the collaborative framework. You don't do this by yourself. These are the lions across the globe, but also particularly in the Netherlands and in Europe. So if you try to think about psychiatry and how certain mental disorders may be initiated, I think it's quite easy to see that you have to take into account the complexity of the interaction between genetic and environmental factors. And this is illustrated in these two figures. On the left-hand side, you see an inverted U-curve where the expression of a certain phenotype is normally distributed in the general population. A specific individual would have a particular genetic liability that is either low or high, and thereby may be of increased risk of developing this illness, which is only in this bottom point of this inverted U-curve. Now, the field of epigenetics and something that is linked ecogenetics may take into account that it's not only about genes or about your environmental liability, that is depicted on the right-hand scheme, but it's about their interactions. And it may be that while your genetic liability may be low, due to your environmental exposures during early life or during later life, you are being pushed against becoming more at risk for becoming ill, and thereby via the alterations of the structure or the expression of specific genes in specific pathways, most likely in the brain, but maybe also involved in the immune system. And vice versa, you can also think about that the environmental liability, when people have a specific genetic background, may be expressed differently, and so that with a specific genetic background, you're either more sensitive for specific environmental exposures, or you are driven towards specific environmental exposure. This is called gene-environment correlations. So the talk about my talk today will be particularly about the field of epigenetics, and Dr. Sinan Guluxus will particularly emphasize the field of gene-environment interaction research. I want to bring you back to biology and look with you what epigenetics is from the more molecular biological background, and that is that these molecular mechanisms are capable of moderating and modulating the effects of environmental exposure, and they are mediated via things like small methyl groups that may attach to the DNA helix, or small modifications that are taking action on histones and histone tails, which may open or close the accessibility of the DNA helix. Or it may also be that certain specific small RNA molecules, they may also inhibit the expression of specific genes. And these mechanisms are under the influence of genetic backgrounds, since specific genetic variants may also give rise to specific levels of methylation, but they also are sensitive for environmental exposure. So this is really truly where nature and nurture meet. Another simplified scheme, but it's coming from DNA to messenger RNA to proteins, and from proteins you have metabolites. The epigenetic mechanisms, they alter the way that genes are being described. The transcription of genes is altered by methylation of DNA and alterations to histone tails, while the expression of messenger RNA or the translation from messenger RNA to proteins may also be influenced by these very small RNA molecules that are non-coding into proteins, but they inhibit the expression of this of other messenger RNAs. And these things are called microRNA. A very interesting metaphor, I think, I like to use some kinds of symbols, is coming from the neuroanatomist Ramon y Cajal. He's a very famous scientist. More than 100 years ago he worked in Madrid, and he drew very nice morphological pictures of neurons. And I in my PhD, I was very much a fan of his work, reading all his books and how different neurons would look about. And he also described neurons are the butterflies of our soul. I think a very nice quote. And if you think about it, butterflies have a specific developmental trajectory whereby they have a very strong phenotypical change coming from a caterpillar into a larvae, into the butterfly. This is a very nice metaphor what epigenetics is and how the environment may alter epigenetic pathways, since the butterfly and the caterpillar had the same genetic background. Due to developmental programming under the influence of environmental cues, the caterpillar is being changed into the larvae and into the butterfly due to epigenetic regulation of gene transcription. So how can you study this field of this method of this process of epigenetics? And I will give you some examples during my talk. I will try to simplify some of the work so that you can fully understand what is being done. So it's a bit simplistic sometimes, but I hope that you will understand. So you can understand, you can measure, I have to go back. So you can measure specific epigenetic relevant genes. So these are genes that code for proteins that are very important for these epigenetic processes. I will come back to this in a minute. This is done by, so you can measure the genetic variance in these epigenetic relevant genes by genotyping. This is depicted here to the left. You can also mark specific epigenetic profiles in a tissue section. So you can have a certain antibody that recognizes a methyl group or specific histone tail alteration, and thereby you can see in sections what type of level specific methylation processes may have in specific cells or specific regions. And you can also measure the levels of the epigenetic profiles themselves in a gene-based or even a smaller CpG-based manner. So you can measure millions of different loci across the genome for how much methyl groups are attached to this to the sequence. And this is done by molecular biological readouts that I will also show you later in the talk. So first, the field that we started with is, I think almost 9 years ago we had the study design done, and then we performed the studies and published in 2014 and 15 about genetic variants that are relevant for DNA methylation, so the addition of methyl groups to the DNA helix. And this is being mediated particularly by these enzymes, MTHFR and DNMT1. So DNMT1 stands for DNA methyltransferase 1, and you have other versions called 3A and 3B. And MTHFR stands for methyl tetrahydrofolate reductase. So it's an enzyme that is reducing the folate levels from specific regions. And you can measure the genetic variants by having specific SNPs that cover the variants within these genes. And some of these things are also very functional, we found out. So we measured these genetic variants called single nucleotide polymorphisms in these genes, and we tried to correlate these things to specific phenotypes. So the phenotype that I was particularly interested in were, of course, mental disorders. So we first did a systematic review of the literature and a meta-analysis, and we found that genetic variants in this gene called MTHFR, they increase risks not only for one specific disorder, but for a range of disorders. So there's a contributing to the shared genetic vulnerability across mental disorders. Not really to a huge effect, but they did. And secondly, we also wanted to test, are these genetic variants also associated with other types of phenotypes? Like, for example, this one. This is a very sensitive one, so you can do experience sampling methodology, meaning that with a specific device, and nowadays we have an app on your iPhone or on your Android device that is measuring 10 times a day for 6 or more consecutive days what you're doing, how you're feeling, and so forth. So you have really nice patterns of all types of emotional stability, but you can also have you can also do cognitive phenotyping by using this type of digital phenotyping. And we had 2 phenotypes that we investigated at that time regularly, and these are very relevant, I think, for psychiatry. And the one that we investigated in this study is particularly the level of negative affects, so negative emotions, in response to events that people perceive as being stressful. So normally you would have a specific variation of your negative effects in daily life. Not many people are very, very stable. Everybody is showing some kind of fluctuation. I thought maybe people with high stress sensitivity, they show a high increase in negative effect after stressful events. Maybe we see this, that specific carriers of the genotypes that are relevant for these different genes, maybe they show different responses in stress sensitivity as you measure these with this phenotyping approach. And we found, in fact, that variants in DNMT3A, don't want to show all the details of the data, but we measured this in 5 different cohorts, and in 3 different cohorts, particularly in general population cohorts and cohorts with minor depressive events, symptoms in people. We found that variations in this gene, DNMT3A, were very relevant and strongly associated with the amount of negative effect after exposure to stressful events in daily life. So this was interesting, we thought. It was a long time ago already that we did these studies, but we wanted to continue this in the field of aging and dementia. This is the field where I did my PhD. First we did some systematic reviews and so forth, and trying to summarize what is happening in the field and trying to distill out what we wanted to do ourselves. And we thought, okay, let's go to a phase before dementia is really apparent. And this is the phase where people have mild cognitive problems. They have also impairments, but they're not really fully socially dysfunctional so that they are being diagnosed with dementia. This is called MCI, mild cognitive impairment, and these are the people that go to the memory clinics and say, hey, I noticed with myself that I have some complaints. We also see them back in the test, but they don't have any dysfunction due to this, but am I at increased risk for later dementia? Is this predicting something that I will decline further in cognitive functioning? And this is, I think, clinically very relevant in a high-risk population. And we wondered maybe if we also take the same genes and the genetic variants, maybe people with specific variants in these genes show a stronger cognitive decline over time. And we had the capacity with our network locally and internationally that we could bring together 170 subjects with MCI, and we had a long follow-up of 2, 5, and 10 years of time. The cognitive testing was being done, and we found that in the different genetic variants across these genes, we found that 3 variants in this gene, again DNMT3A, were associated with cognitive decline over time in people diagnosed with MCI. And we also replicated this in another in a replication cohort which was larger in sample size, but the follow-up assessments were done at 1, 2, and 3 years, and we found that one specific genetic variance was also showing the same effect, same direction of effect, but very small effect size. And these things need to be replicated. And nowadays people are mostly working with polygenic risk scores or GWAS-based studies. And this phase, this was still, I think, very relevant. But also, I think, we were really doing this for a hypothesis testing study. So this gene called DNMT3A seemed to be of interest, and during my PhD I did an animal study. So I'm a psychiatrist who has a PhD in experimental animal studies, and we did a very large aging study with mice, looking at the effects of antioxidants and caloric restriction, how this may impact on health, lifespan and also on health of these animals. And we were lucky to be able to follow them at specific time points, 6 months, 12 months, 18 months, 24 months. And then some of the groups were already almost completely deceased, and 2 groups were still living. So these were the animals which were kept under a diet of caloric restriction, which is known to increase maximum lifespan. But you can also see that during a PhD you have certain sensitive days, and this day suddenly all remaining animals died. I was at a conference like here, I was receiving a phone call, and somebody in the lab said, Bart, all your animals died on one specific day. This was a big disaster for the study, since we couldn't follow up and see the difference between these 2 groups, since this group seemed to be aging better. But also shows that how sensitive these experimental animal studies are. And I'm still not sure what happened during the day, whether the water was poisoned or something else. But I was lucky, since just a few months before that time, I took out a group of animals. So I still could do a comparison study comparing animals aged up to the age of 24 months, so 2 years, which is quite long for small animals as mice are, and 12 months of age. And we did some immunohistochemical stainings of marks that are associated with DNA methylation. And the first one we did is giving our genetic findings on DNMT3A. We want to see, is this protein expressed in cells? And yes, it is. In almost all cells, this protein is being expressed. And you see this in the hippocampus of an animal. This is work being done by Dr. Goudiaris, who is now a psychiatrist working in Cambridge. And he did the study and he found, okay, this protein is being expressed, but in the dentate gyrus, this is expressed by specific cells to a much higher extent. And in other genes, in other areas, he found a more ubiquitous expression. And if you would do the aging study, what he found is that with aging, the level of DNMT3A increased. These are these groups, wild type, control diet. You see that with age, there's a small increase of the expression level of this gene, of this protein. But you also see that with caloric restriction, this age-related increase was not really observable anymore. So there was a aging times caloric restriction interaction. Next, we also looked at these cells with a high level of expression, and these were identified as newly born neurons. So we also injected them with BRDU, which is a marker for cell proliferation. We sacrificed some of the animals a few weeks later, and then you see that animals that had BRDU in their nucleus also had DNMT3A in their nucleus being expressed to this high level. And this is, I think, also still quite logical, since if you have newly born cells, they also have to re-establish all the methylation profiles. So therefore you need high amounts of this protein to do this. So this type of cells, they decreased with aging. So, and it's also known, I think, that with aging there's a loss of the neurogenesis or neurogenic capacity in the hippocampal dentate gyrus. We also found in the same animal model that with aging, not now looking at DNMT3A, but at the product of this, so the methylation of cytosins in cells, you can also have a specific antibody recognizing this, and you see that all cells are stained, and that with aging, these are 12-month-old animals, these are 24-month-old animals, different hippocampal regions, you see a strong increase of this staining intensity, which was again attenuated by caloric restriction. So animal studies are very interesting, but of course you want to know how does this relate to human beings, and is there a relevance for this, for example, in the field of dementia. So we started to collaborate with a very nice research institute in the United States. We had the same Greek scientist, and is now a psychiatrist, who we sent to the United States to do human experiments with post-mortem material. And we sent them to this institute since they have a wonderful follow-up of people living in the elderly communities in Phoenix in Arizona, and they have ante-mortem phenotypes. So before people are being diagnosed with dementia, they have blood samples available, they have clinical phenotypes, and they follow up these people. And upon death, they have the informed consent also to collect some of the tissue. And what you can see is that the average post-mortem interval of this entire brain bank, of all the brains collected in this community, is 2.8 hours. So within 3 hours of time, the brains are being dissected out and being preserved in buffer, which is, I think, very useful for doing proper research. So what we did is we stained the sections of people with dementia and control populations of age-matched controls. And what you see here is that with stainings for, again, 5-methylcytosine in the hippocampus of human beings, you see that the control population, you see a very strong labeling, which is lost in people with dementia of the Alzheimer type. And this suggests, or this is quite similar to what was also found out in these animals, and we also found similar things in transgenic animals that overexpress mutant forms of APP and PS1. So we now are following up this in the sense that we're not doing stainings anymore, we're now measuring the methylation profiles in specific genes with array technology. So what you need to do is to extract the DNA from tissue and cells, You have to convert the DNA into a little bit other molecules that you can measure better in the molecular measurement machineries. But what you can then do is per DNA sample, you can measure more than 500,000 or 450,000 loci across the genome and measure how much methylation is present in specific DNA samples. The limitation of this technique is what we normally do is take homogeneous of tissue. So you don't have any cell type specific information. You have just a chunk of hippocampus or a chunk of cortex or a chunk of middle temporal gyrus sample, and you measure what is in there. And you can, with methylation data, you can also try to correct for cell type composition, but it's still homogeneous. And first papers were already published, and we're now in the process of trying to publish our paper where, with an international group of people, we're looking at DNA from the middle temporal gyrus and compare normally aged people versus those with AD, and we also have people with different stages of neuropathology, the Bragg stages, so we can also compare across these pathological stages how the methylation profiles are being changed. And just to cut to the chase, so these are the data which I won't discuss with you in detail, but these are some Manhattan plots on the p-values of those findings on 5-methyl-C, and this is the level of genome-wide significant detection. And at the single loci level, we didn't see anything that exceeded this level of statistical significance. What we did find is that specific regions where multiple loci in a specific gene would show a similar effect, that they show quite striking differences. And these were differences were to be found in specific genes. And a few of them were already mentioned in previous papers, complement factor 3, and a new one was found in oxytocin. So what we found is that oxytocin at the level of methylation and also at the level of expression is altered in people with dementia and also compared to age-met controls. What I didn't put into this presentation is that we also nowadays have blood samples isolated from people with MCI, and they also, people have the methylation of the oxytocin gene at baseline when they have MCI is predictive of later conversion to dementia. So I think that's quite an innovative finding how oxytocin is linked to neurodegeneration. Okay, that's our work on dementia. And now I would like to shift to my prime research interest over the last 5 years, and this I think very relevant for the broader field of psychiatry is the link between trauma and psychopathology. And for doing epigenetic studies in human cohort studies, I wanted to tap into the biological mechanism, so you need to have biological tissue. And for people exposed to trauma, or people with PTSD, there's no human brain bank collection. So you don't have post-mortem material where you could investigate differences. And you don't also, I think, what is very worthwhile is what you would like to do is to measure changes over time in the same individual. So ideally, you would have people that are either sick and then are recovering, or you would have people that are not sick and then are exposed to something whereby some of them are developing a mental disorder. So in the first pilot study that we did, or proof of principle study, I wanted to see, does it make sense to measure epigenetic profiles in blood samples? Since if you don't find anything that is meaningful, then this is a very difficult area of research. So I wanted to have this paradigm where there's a change in the phenotype in the same individuals, and that we could measure things before and after a specific change in the phenotype. That was also particularly being induced by an environmental factor. So I talked to our department chair at that time and said, Jim, I would like to do research on ECT, electroconvulsive treatment. Since these people are admitted to the hospital, they're all quite sick, treatment resistant, and it's the best or the most effective treatment strategy we have in psychiatry, and we expect changes in the phenotype. And I'm wondering, can we also detect something meaningful and that correlates to the changes in symptoms that we can measure in blood samples? So we did. And in our Maastricht-based hospital setting, we did an ECT study, a prospective one where people, approximately 20 people that came into the hospital, were followed up on a weekly basis, received electroshock treatments, where we collected blood samples on a weekly basis, and also phenotyping on depression scores, but also on cognitive functioning. And what we found was that, of course, the depression scales, they responded to ECT treatment. Here you see the treatment response in the Hamilton rating scale, or in the back depression you see during treatment the symptoms are going drastically down, and after the treatment has stopped, in the follow-up period, people remain stable. And you see a little bit of a tendency to show increased depression scores in the BDI, which is a self-report scale. Interestingly, and that's what I wanted to see, I was hoping to find in peripherally circulating PBMCs, so blood cells. We took the messenger RNA, we isolated this, and we measured specific genes we thought that could respond to ECT treatment. And this is brain-derived neurotrophic factor. This is expression levels at the messenger RNA level in blood cells. So in circulating blood cells, you see during the treatment period that these respond. They increase expression of BDNF maybe as a consequence of ECT. At least it's quite nicely showing this during the treatment period, and after the treatment period nothing is altered anymore. So this was thrilling to me. It's not still unpublished, but so BDNF changes in blood cells in response to ECT, and they also strongly correlated to the antidepressant response to the Hamilton, the MADRS, and the BDI rating scales. So it does make sense. It may make sense to measure blood samples and measure expression of genes or methylation profiles in these types of studies. Now we also did a methylation analysis, and we found a specific loci across the genome. They also show differences or alterations in response to ECT. So this really supported my ideas of studying this. And then I thought, okay, what is the most relevant environmental risk factor in psychiatry? This is, of course, trauma exposure. And I wanted to do a prospective study where people were followed up after baseline, and you had follow-up measurements. You would have data on the clinical phenotypes before and afterwards, and you would also have blood samples available. So I thought, where can I get this type of human cohort? So I went to the Ministry of Defense in the Netherlands, where I knew Professor Erik Vermetten. He's a psychiatrist. He's coordinating these type of studies. And in the Ministry of Defense, there is cohorts of military soldiers being deployed to combat zones. So there's one very nice epidemiological study in the Netherlands done. It was started in 2004 up to 2009, and it's still continuing since we're now doing a 10-year follow-up and even longer, where more than 1,000 subjects, so military soldiers, were deployed to Afghanistan, were phenotyped, blood samples were collected before and after the deployment period. And for my first study on epigenetics in this cohort, we chose to compare samples of blood and DNA that's of 6 months after the deployment period and those at baseline. So these things are quite pricey. So one array of methylation costs about €350. So if you want to do 2 per individual, it's about €700 for one individual, times 1,000. So this study was really not feasible financially to do this for the entire cohort. So I selected a few sub-cohorts, so smaller cohorts that were polarized or showing a polarized phenotype. So 32 individuals each per group, 2 time points, and these people, I call them GYM. The GYM group, they changed over time in the sense that they developed mental ill health. They developed PTSD over time once they were exposed to deployment-related stress and trauma. Another group was not showing this change. They were exposed to the same level of stressors but remained healthy. So this is more the unsusceptible or resilient group. And a third group was not really exposed to the combat zone since they remained in base camp. They were still in the same region, they had the same diet, the same population, homogeneous men mostly that are fit and healthy, not exposed to trauma as a third and a second control group. This was done in collaboration with Erik Vermetten, with Marco Box in Utrecht and other people in Utrecht, but also with a United States-based cohort where we replicated our findings with Doreen Baker and Caroline Nievergeld. So what we did is, before and after the deployment period, we collected blood samples. We also clinically phenotyped them. We measured the level of DNA methylation in more than 450,000 loci and the changes of them over time. And we measured the changes over time in the symptom scores of PTSD. And then we tried to link these two to test whether changes in PTSD symptom scores would be associated with specific changes in DNA methylation. Again, Manhattan plots where we found now genome-wide significant hits in specific genes and also regions that were showing a statistical significance. And these were in specific genes that we now are following up upon in our laboratory studies. We also replicated 2 of the regions, and these were found in regions of the gene RNF39 and a gene called zinc finger protein 57, ZFP57. Another interesting thing I think what you can do with these data, you can go back to the individual, look at the level of methylation before and after deployment, and see whether they, particularly also in the second group, how the individuals change over time in methylation level. So this is one of the genome-wide significant loci, a locus in DASP22, a specific gene. What you can nicely see here in this plot that this is time point baseline, time point follow-up after 6 months, that the individuals show quite robust changes. There's a group that's particularly linked to a specific genotype that does not really change, but these people all show quite substantial changes. And people that are in the susceptible group, they develop PTSD, they all tend to go down in methylation level, while the people that are resilient go up in methylation level. I think this is a very interesting finding as well. It suggests that resilience, so coping with adversity in a longitudinal manner, is linked to active biological processes that you can measure in peripheral samples. I think that's quite cool. So resilience is something active in a biological perspective. We also did some further computation analysis with bioinformatics and so forth, where you can test for enrichment of specific signals in your findings that should be linked to specific organelles or biological processes. And here we tested this, and we found in these blood samples that the DNA methylation profiles that were coming out as the most statistically significant were mostly enriched in a specific organelle of cells, which you would expect to be blood cells or immune cells, but this was neurons, neuron part. So number 1 in the GO categories of the cellular component was neuron part, neuronal projections, which is quite amazing, I think, to find this in such a interesting paradigm. Also, and this is I think quite innovative in the sense that normally people would do, in epigenetics, do a more targeted hypothesis-driven approach, that they test the glucocorticoid receptor or something else. This is an agnostic approach of looking at the data, finding in the data which type of biological processes would be best linked to changes in PTSD symptom scores. What we find were biological processes that are quite relevant, that people would already think about that would be relevant. And this is amazing, I think. So what we see here is that the enriched, most highly enriched pathways or processes were these. This is the sudden infant death syndrome susceptibility pathway, mostly serotonin, circadian rhythm, IL-17 signaling pathway, and Professor Brian Leonard here in the audience will be excited by this. But also here the transporter of serotonin, the TSH signaling pathway, and so forth, sleep regulation, complement activation, so all types of biological processes you would hope to find, you can also find back in these data, which were collected in an agnostic manner. So I already told you we did a discovery data set in the Dutch military, we did a replication in the US data set, and these are the data of the regions in zinc finger protein 57 and RNF39. And this is people with an increased PTSD score, so the people that are susceptible, these are the people that are resilient, and they show quite opposite directions of effect. In this, and the same directions apply to both of these data sets. So what about these genes? RNF39 is ring finger protein 39, and ring stands for really interesting new gene, interesting name for a gene, I think. It's involved in the shuttling from the synapse to the nucleus. It's also known to be involved in the early phase of synaptic plasticity in LTP. It's activated upon fear conditioning, and is also upregulated upon stress in the hippocampus of animals with stress during earlier life. So kind of sensitization of stressors, which you can also think about maybe something that is in action in these military soldiers. For the first time, we didn't find any immunohistochemical findings, so how this protein is expressed. This is a picture that we did with staining for a human antibody for ring finger 39 is on the human hippocampus, and this is the mouse hippocampus showing quite striking hippocampal staining. Zinc finger protein 57 is the other interesting gene that we picked up and identified. It's a transcriptional repressor. It's essential for genomic imprinting, and genomic imprinting is necessary in normal development, so it may impact on a range of other genes as well that are genomically imprinted. It's a binding partner of another protein which mediates, again, fear conditioning. It's folate sensitive, but there's limited knowledge of this in the brain, so we also took an antibody, we stained it, and we co-localize it with NOIAN, so it's present in neurons in the brain. We don't know what it's doing there particularly, or if you manipulate it, what may happen. So then we thought, okay, this is interesting, the human findings. Can we also validate this in an animal setting? So I collaborated with people in New York, and now Nikos is in Harvard. Nikos Daskalakis, he did an animal study on different types of behavioral phenotypes that he found in the predator scent stress model in rats. And he did this by grouping the animals based on specific behavioral phenotypes, not only one behavioral phenotype, but multiple behavioral phenotypes, and say these are the susceptible animals, these are the resilient animals, and we could compare these. And what we found, if we would test these animals for expression in blood samples of our genes that we found were of interest, that there was striking correlations with the startle response in these animals, with startle habituation, with time spent in the open arm, so all types of phenotypes that would be associated with PTSD-like behavioral phenotypes. So this, I think, supports the idea that these things may be relevant for PTSD. Next, we also collaborated with other people at the Libre Institute. We thought, okay, if we have these 450K methylation data, I also knew that other people have the same type of data in other data sets, and particularly in post-mortem brain material of people with depression, bipolar disorder, and schizophrenia. The group of Daniel Weinberger and Joel Kleiman has collected these, and Luca Orsini is a postdoc that I met at a conference like this. We went running in the park together, and then we talked about science, and he said, oh, but we have these data sets. Don't you want to test your data, your findings in our data set? And I said, yes, we should. And we found that, so in our PTSD code, we found hypomethylation over time in people that were susceptible in PTSD, so lower levels of methylation. And this was also something that we found in people with schizophrenia, and particularly schizophrenia in RNF39, and also for zinc finger protein 57. Now this is post-mortem brain material of people who've had long-term mental illness, medication, and these things. So I don't want to imply that this is due to traumatic events or so, but it's still a quite interesting thing that they were hypermethylated also in this data set. As a last point, before I think we can go towards the conclusions, is that I wanted to mention that we're now also looking at another epigenetic process called these microRNAs, the non-coding RNAs that inhibit the expression of other genes. And the interesting thing about these things is that they can circulate, so they can be produced in the brain and the central nervous system. And some of the cells, or all of the cells, they make exosomes or microvesicles which can contain these microRNA species. And then they can go into the cerebrospinal fluid, go into the blood circulation, be detected and be isolated in the blood samples. And now we have tools also to do this. So you can isolate exosomes or microvesicles that are most likely derived from the central nervous system based on certain receptors on the membrane. And you can then pull them down and measure the microRNA content in these vesicles. And this has been done. Our postdoc Laurence Denys has been doing this, but only since we had limited financial resources. For this stage, we have done this for 24 samples after the deployment periods, where we found very striking differential expressions of these microRNAs. And this is still work in progress. We're now trying to replicate this in the US-based cohort or in an animal study and submit it soon, since we found quite remarkable differences. So out of the 918 sequenced microRNAs, I think more than 200 were differentially expressed between the groups. We have to validate this and replicate, but I think that's a very interesting first discovery which should be tested further. What we're also doing now, I think there's different levels of biology. I already mentioned in the beginning, there's a genetic background, there's an epigenetic profile that can be imprinted by the environment. This will translate into messenger RNA. Messenger RNA molecules are then translated into proteins and then metabolites. And the best thing to do is to try to build a type of data set where you have all these biological levels measured in the same individuals at the same time points. So we are now doing this. We have data on the genetic background of all these individuals in the Dutch military. We have methylation data in about 96 individuals, 2 time points. We have microRNA expression data. Now we are doing this in a larger sample than these 24. Just last week we have finalized the messenger RNA expression data. We have data on protein and proteomics, and we have data on metabolites by collaborators in Zurich and in Ireland that were so interested by the study that they say, okay, we can do this for free. And now we are in the difficult situation that we have to integrate these levels of biology. To try to harmonize the data sets, that's one thing, and then to make a biological meaningful story about what may happen across these biological levels, and also how we measure them in peripheral blood samples. So that's, I think, where we stand. So there's some other further developments. I really wonder about cell type specificity. Even in blood samples, there's different cell types. And what we didn't do in the previous military study is we didn't really sort out the specific cell types. Now you can do this, but at that time the study was not designed in this manner. So I think what we are doing now, and what we should do more, is if you collect blood samples, isolate the specific cell types, and then store them independently so you can do sequencing separately on different cell types. What we also try to do is to isolate circulating stem cells or get bone marrow-derived stem cells, since the blood-based DNA methylation changes that we found were 6 months after the deployment period. So these blood cells, they are being replaced every 2 to 3 weeks. So these blood cells that were present at the moment of investigation were not present at the moment of exposure. And this suggests that something may also have imprinted the bone marrow stem cells that may then have been proliferating and giving rise to a little bit different blood cells that have a specific DNA methylation profile. So we also contribute to larger psychiatric genomics consortia initiatives, such as that one in PTSD. We want to replicate further, validate, and also, as Professor Taminga just showed out, what we're doing now also is experimental studies, not only in animals, but particularly also in cell models. So I got a nice grant to do iPS cell reprogramming and then neuronal differentiation in the same subjects of the military cohort in the Netherlands. So we have living neurons of the individuals that we previously had these blood-based data on. And I think that would be a very interesting opportunity to test what we see in vivo. Is it also reflected in vitro in cell models? And for this, we're now establishing stress models or stress exposures in a dish, which is also quite an interesting exercise, not fully finalized yet. So tomorrow, Dr. Sinan Guluxus will talk about gene-environment interaction in psychosis. What he will not talk about is that we're also doing DNA methylation studies in these cohorts. I just wanted to end with this conceptual framework where I think over time, with development, prenatally, perinatally, and also during adolescence, there's developmental changes that should not be looked upon in diagnostic categories, but mostly, I think, in dimensions of symptoms and clusters thereof, and networks of symptom clusters, and also how they change over time. That would be relevant. I think that we can really improve much better. And then also then integrate the measurements of epigenetic profiles or other levels of biological profiling using peripheral samples. But this should be done in a longitudinal manner. So just to conclude, the environment matters, I think, literally. We should do long-term longitudinal studies. There should be more crosstalk between epidemiology and neuroscience, I think. We should think about cell type specificity and so forth. But also have to say what I also foresee is that we need really much more collaborations nationally, internationally. So I think our network is very open to people from Turkey or other places to work with us on this exciting endeavor. So with this, I would like to thank you for your attention. I'm open for your questions. Thank you.